So I'm Jeremie. I'm working in the lab of Alex Reyes at the Center for Neural Science at New York University. First of all, I would like to thank the organizing committee for giving me the opportunity to present my work. And we're going to go back a bit to experiment, not to the brain, but to something in between theory and experiments. So I'm studying how signals is propagating in the feedforward networks. So feedforward networks are a general concept in order to understand how an input is processed into a multi-layer network. And you can think as the auditory pathway as a feedforward network, or the thalamocortical pathway, where thalamic input excites successively the different layers of the cortex. But in ideal feedforward networks, neurons only make connection from one layer to the next. There is no recurrent connection. And this type of network has been extensively studied using theoretical tools. And people have studied in particular which are the conditions to either propagate an input or what are the conditions where the input, the activity is stopped. And there are two main parameters that will determine the dynamic of the network. First, there is a structural parameter, so the connectivity. What is the strength of the connectivity, whether there are divergent and convergent connections, and also the number of neurons per layer. And then there is a functional parameter, which is the input. A synchronized input is much more likely to propagate in the deepest layer than an asynchronous one. And so these theoretical predictions are very hard to test in a biological network because it's hard to isolate feedforward networks in vivo. And so the goal here of the experiment is to develop an experimental preparation where we could study feedforward networks in biological with biological neurons. And so we will develop an alternative strategy and we'll use the multi-layer culture preparation that I will describe now. So the idea is to use a culture of network, of neurons in vitro, and I use microfabrication techniques in order to build these compartments where neurons will grow. So here you can see a microscopic view of the network. And so the neurons only grow in these rectangular chambers. And after 2 days in culture, neurons will make recurrent connection within a layer, but will also extend long-range connection between layers. And I will use patch clamp recordings to monitor spiking activity in the different layers in response to optogenetic activation of the first layer. And so in order to control faithfully the inputs that I deliver to the network, I will use pattern optogenetic stimulation. So here you can see the microscopic view of the network. Excitatory neurons express channel rhodopsin, so we can label them, and we can record from neurons that don't express channel rhodopsin to avoid any cross-activation. Then I build a setup based on the working of a digital micromirror device that enable me to project any pattern of light with a very high spatial and temporal resolution. So I can now draw region of interest onto the neurons that I want to stimulate from the first layer, and I can project any pattern of light. And so here I'm showing 3 examples where each neuron is stimulated with a single pulse, and I'm varying systematically the temporal precision of the input. So now I'm going to show some experimental results. So here is an example of propagation. Each line here in this raster plot represents the repetition of the exact same stimulus. And so you can see that the synchronous activation of the first layer evoked a small activity in the first layer, a transient activity which is propagated along the network. In the second example, a similar activity, so it's a different network, a similar stimulation evoked a transient activity which is then followed by a long-lasting persistent activity. And so you can appreciate that here, that this transient activity disappeared after layer 1, but only the transient activity is well propagated. And the main difference between these two networks is that they differ from their density. On the left side here, you have a sparse network that contains a low number of neurons, and on the right side, you have a dense network with many neurons in each layer. And so these two networks behave differently. We could show that this sparse network behaves like a thin fire chain by measuring the delay of propagation, the delay of propagation is in agreement with activity propagating only through feed-forward connection without any recurrent connection. In the case of the dense network, activity propagates with a slower speed and with a high firing rate. So we name this mode of propagation the rate mode. So to further characterize how activity propagates in this network, we draw the phase diagram of spike propagation. And so here I'm going to plot the probability of spiking in each layer as a function of the spread of the spike. And so the input here is a synchronous input, which is represented here. And as activity propagates from layer 1 to layer 4, probability decreases, so propagation stops. And we observe a small enlargement of the spread. And so to plot the full phase diagram, we varied the temporal jitter in the input, and we could draw all of these trajectories. And this phase diagram resembles the one that is predicted by theory, where propagation fails and the number of spikes decrease as a function as propagation occurs. So this is the phase diagram predicted for a purely feed-forward network. But this work also predicted that if we were able to increase the number of neurons per layer, the network state could reach an attractor where where activity could propagate indefinitely with a very sharp temporal precision. So we increase the number of neurons in our network by increasing the density of the cells. But we observed a completely different behavior. The probability of propagating was much higher, so the network was able to propagate reliably, but the spread was increasing as propagation occurred, which suggests that the mechanism of propagation is different. And indeed, we observed that the transient activity was lost, but propagation occurred through this persistent phase, which is different from the one that is predicted by theory. So in the next part of the talk, I will now study what signal is propagated via this persistent activity. And so the first hypothesis that we had is that the network can propagate firing rate. And so to demonstrate that, we sorted our data according to the firing rate in the first layer, and then we measure what is the firing rate in the deepest layer. And so low firing rate in the first layer resulted in a low firing rate in the deepest layer. But increasing firing rate in the first layer resulted in an almost proportional increase in the firing rate in the deepest layer, which suggests that this network are able to propagate firing rate information. And so to further demonstrate that, here I'm plotting the firing rate in a given layer as a function of the firing rate in the previous layer. And we measure this monotonic and unique relationship, which shows some sign of saturation at high rate, but at low rate, it is almost linear with a slope 1. So for a small range, this network are able to propagate firing rate information faithfully. But this network are able to propagate firing rate information, but they're also able to propagate temporal information. And to demonstrate that, we varied systematically the temporal dispersion of the inputs. And I'm going to plot a raster plot and firing rate of the neurons in the different layers. So here is the response of neurons in the 4 different layers. in response to a synchronous input. And when we increase the temporal dispersion in the input, we observe that the firing rate was decreased in the four layers that we recorded here. So which suggests that the firing rate in the different layers can provide some information of the stimulus. And so to characterize that, we calculated the mutual information between the stimulus and two features of the response. First, we use the timing of the first spike as a way to decode the stimulus. And so we could measure some, we had some decoding accuracy in the first two layers, but then mutual information dropped to chance level after layer 2. Then we use the firing rate during this persistent activity as a way to decode information. And so we had a smaller mutual information in the first layer, but then it remains fairly constant. And when we use both features to decode information, we got a slight increase in the mutual information. So now we have a higher bound of the mutual information, and we can calculate how much information is either contained by the spike timing or by the firing rate. And so what we observe is that the spike time is more informative about the stimulus in the first layer, whereas the firing rate is more informative about the stimulus in the deepest layer, suggesting that this network is able to transform temporal information of the stimulus into a firing rate code. So to conclude, I've shown that we observe two types of propagation. Sparse networks are working like a thin-fire chain, whereas dense networks are working with a rate mode. And with this rate mode, networks are able to propagate firing information. And they're also able to transform temporal information from the stimulus into a firing rate code. So with that, I would like to thank the people I work with, and in particular, Alex Reyes, my advisor. I would like to thank funding and you for your attention. Thank you.
We have time for some questions.
Yeah. So synaptic reliability in vivo is a lot lower than in vitro generally.
Synaptic?
So synaptic reliability in vitro is a lot higher than in vivo. And I was wondering if you know how robust your results would be if, for example, by having a lower calcium concentration, you had a lower synaptic reliability in your network.
So, no, I don't know. So we know that the strengths of the synapse are important, and we know that this sparse network tends to form very strong synapses, and so that's why they're working like a thin-fire chain, whereas dense network form smaller synapses, but I don't have any insight about the reliability of the synapse themselves.
I just had one question. Are you able to record from long enough timescales to see long-term plasticity in these experiments? And do you see any effects of that?
So yeah, that would be a follow-up of this experiment to study, maybe to try to teach them something, right, or that would be. But for that, we would need to record for a much longer time and to have a different setup.
All right, let's thank the speakers again.